A warm greeting to you all. I'm Mrs.Maryann, working as a tutor in Department of Fundamental of Nursing from Sri Balaji College of Nursing. Today we are going to discuss about enzymes from biochemistry. Let's see in detail about enzymes. Introduction. Enzymes are biological catalysts that speed up the rate of the biochemical reaction. Most enzymes are three-dimensional globular proteins, tertiary and quaternary structure. Some special RNA species also act as enzymes and are called ribozymes. Example, hammerhead ribozyme. Structure of enzymes. The active site of an enzyme is the region that the substrate, cofactors, and the prosthetic groups and the residue that helps to hold the substrate. Active sites generally occupy less than 5% of the total surface area of enzyme. Active site has a specific shape due to tertiary structure of protein. A change in the shape of protein affects the shape of active site and function of the enzyme. Active site. Active sites can be further divided into binding site, catalytic site. It chooses the substrate and binds it to active site. Catalytic site. It performs the catalytic action of enzyme. Cofactor. Cofactor is the non-protein molecule which carries out chemical reactions that cannot be performed by standard 20 amino acids. Cofactors are of two types: organic cofactors, inorganic cofactors. First is inorganic cofactors. These are the inorganic molecules required for the proper activity of enzymes. In examples, enzyme carbonic anhydrase require zincs and hexokinase as cofactor magnesium. Organic cofactors. These are the organic molecules required for the proper activity of enzymes. Example, glycogen phosphorylase requires the small organic molecule pyridoxal phosphate. Types of organic cofactors. We'll see about prosthetic group and coenzyme. Prosthetic group. A prosthetic group is a tightly bound organic cofactor. Example, flavins, heme groups, and biotin. A coenzyme is a loosely bound organic cofactor. Example, nicotinamide adenine dinucleotide. An enzyme with its cofactor removed is designated as apoenzyme. The complete complex of a protein with all necessary small organic molecules, metal ions, and other components is termed as holoenzyme of holoprotein. Substrate. The reactant in biochemical reaction is termed as substrate. When a substrate binds into an enzyme, it forms an enzyme substrate complex. When substrate joins in enzyme, it is called as enzyme substrate complex. Next, the sites of enzyme synthesis. Enzymes are synthesized by ribosomes, which are attached to the rough endoplasmic reticulum. Information for the synthesis of enzyme is carried by DNA. Amino acids are bonded together to form specific enzyme according to the DNA's codes. Intracellular and extracellular enzymes. Intracellular enzymes are synthesized and retained for the use of cell itself. They are found in the cytoplasm, nucleus, mitochondria, and chloroplast. Example, oxidoreductase catalysis, biological oxidation, enzymes involved in reduction in the mitochondria. Extracellular enzymes are synthesized in the cell but secreted from the cell to work externally. Example, digestive enzyme produced by the pancreas are not used by the cells in the pancreas but are transported to the duodenum. Characteristics. Enzymes speed up the reaction by lowering the energy of the reaction. Their presence does not affect the nature and properties of end product. They are highly specific in their action, that is, each enzyme can catalyze one kind of substrate. Small amount of enzymes can accelerate chemical reactions. Enzymes are sensitive to change in pH, temperature, and substrate concentration. The turnover number is defined as the number of substrate molecules transformed per minute by one enzyme molecule. So the catalase turnover number is equal to 6 into 106 per minute. Nomenclature of enzyme. An enzyme is named according to the name of the substance catalysis. Some enzymes were named before a systemic way of naming enzymes was found. Example, pepsin, trypsin, and renin. By adding suffix "ase" at the end of the name of the substrate, enzymes are named. Enzymes for catalyzing the hydrolase is termed as hydrolase. Example, maltose plus water, maltase, which gives glucose plus glucose. Example, substrate, enzymes, and products. So first one is the lactose. In enzymes, it becomes into lactase, and in product, glucose plus galactose. In substance, maltose. In enzymes, it is maltase, and in product, glucose. Substrate, cellulose. In enzymes, cellulase. In product, glucose. Substrate, it is lipid. Enzymes, it is lipase, and product, it is glycerol plus fatty acid. In substrate, it is starch. In enzymes, it is amylase, and in product, it is maltose. In substrate, it is protein. In enzymes, it is protease, and in product, it is peptides plus polypeptide. So we'll see the classification of enzymes. A systematic classification of enzymes has been developed by International Enzyme Commission. This classification is based on the type of reaction catalyzed by enzymes. There are 6 major classes. Each class is further divided into subclasses, and so on, to describe the huge number of different enzyme catalyzed reactions. Classification of enzymes. Enzyme class, reaction type and example. Enzyme classes, oxidoreductases. Reaction type is reduction or oxidation, and the example is lactate dehydrogenase. The enzyme class is transferases. The reaction type is move chemical group, and the example is exokinase. And hydrolysis, that is in reaction type it is hydrolysis, bond cleavage with transfer of functional group of water. Example is lysozyme. Enzyme classes lysases. Reaction type is non-hydrolytic bond cleavage. Example is fumarase. Enzyme classes isomerases. The reaction type is intramolecular group transfer, that is isomerization. Example is triose phosphate isomerase. Enzyme classes ligases. The reaction type is synthesis of new covalent bond between substrates using ATP hydrolysis. Example is RNA polymerase. Mechanism of enzyme action. The catalytic efficiency of enzyme is explained by two perspectives: thermodynamic changes and processes at the active site. Thermodynamic changes. All chemical reactions have energy barriers between the products and the substrate. The difference in the transitional state and substrate is called activational barrier. Only a few substances cross the activational barrier and its products. This is why rate of uncatalyzed reactions is much slow. Enzymes provide an alternate pathway for conversion of substrate into products. Enzymes accelerate reaction rates by forming transitional state having low activational energy. Hence, the reaction rate is increased many folds in the presence of enzymes. The total energy of the system remains the same and equilibrium state is not disturbed. So you can see the thermodynamic changes overview without enzyme and with enzyme. So without enzyme, lactose, glucose plus galactose. With enzyme, lactose, glucose plus galactose, activation energy with enzyme. So the energy is not released. So processes at the active site: covalent catalyst, acid-base catalysis, catalysis by proximity, catalysis by strain. So covalent catalysis. Enzymes from covalent linkages with substrate transient enzyme substrate complex with very low energy. Enzyme is released unaltered after completion of reaction. Acid-base catalysis. It is mostly undertaken by oxidoreductases enzyme, mostly at the active site. Histidine is present, which act as a both proton donor and a proton acceptor. Catalysis by proximity. In this catalysis, molecules must come in bonding distance. When enzyme binds, a region of high substrate concentration is produced at active site. This will orient substrate molecules, especially in a position ideal for them. Mostly it is undertaken by lyases. The enzyme substrate binding causes reorientation structure of site due to in a strain condition. Thus, transitional state is required, and here bond is unstable and eventually broken. In this way, the bond between substance is broken and converted into products. So we can see substrate active site, that is substrate entering into the active site of the enzyme. Enzyme changes its shape slightly as substrate binds. It forms into enzyme substrate complex. After that, the enzyme products complex, then the products leaving the active site of the enzyme. First, we'll see about the lock and key model. Was proposed by Emil Fischer in 1894. The lock and key hypothesis assumes the active site of an enzyme and ridge in its shape. There is no change in the active site before and after a chemical reaction. Induced fit model. Most recent studies have revealed that the process is more likely to involve an induced fit model proposed by Daniel Koshland in 1958. According to his exposure of an enzyme to substrate causes a change in the enzyme, which causes the active site to change its shape to allow the enzyme and substrate to bind. That is, this is the induced fit model. Next is the enzyme kinetics. It is a branch of biochemistry in which we study the rate of enzyme catalyst reaction. Kinetic analysis reveals the number and order of the individual step by which enzymes transform substrate into products. Studying an enzyme's kinetics in this way can reveal the catalytic mechanism of an enzyme, its role in metabolism, how its activity is controlled, and how a drug or an agonist might inhibit the enzyme. So the rates of reaction and their dependence on activation energy. Activation energy is the least amount of energy needed for a chemical reaction to take place. Enzyme, as the catalyst, acts on substrate in such a way that they lower the activation energy by changing the route of the reaction. The reduction of activation energy increases the amount of reactant molecules that achieve a sufficient level of energy so that they reach the activation energy and form the product. Example, carbonic anhydrase catalyzes the hydration of molecules per second, which is 10 times faster than the spontaneous hydration. So this is the energy level of molecules, that is the reaction which is seen. The kinetics of enzymes catalyst. Enzyme catalysis, it is an increase in the rate of reaction with the help of a enzyme as catalyst. Catalysis by enzymes that proceed via unique reaction mechanism typically occurs when the transition state intermediate forms a covalent bond with the enzyme. Covalent catalysis. During the process of catalysis, enzymes always emerge unchanged at the completion of the reaction. Factors affecting the rate enzyme catalyst reactions: temperature, hydrogen ion concentration, and substrate concentration. Raising the temperature increases the rate of catalyzed reaction by increasing the kinetic energy of molecules. Enzymes work maximum over a particular temperature known as optimum temperature. Enzymes for humans generally exhibit stability temperature up to 35 to 45 degrees centigrade. The temperature coefficient is a factor by which the rate of biological process increases for a 10 times increase in temperature. For most biological process, however, sometimes the heat energy can also increase the kinetic energy to a point that exceeds the energy barrier, which results in denaturizing of enzymes. Effect of pH. Rate of almost all enzyme catalytics depends on the pH. Most enzymes exhibit optimal activity at pH value between 5 and 9. High or low pH value than optimum value will cause ionization of enzyme, which result in denaturation of enzyme. Pharmaceutical importance. Enzymes are virtually involved in all physiological process, which makes them the targets of choice for drugs that ameliorate the human disease. Applied enzyme kinetics represents the principal tool by which scientists identify and characterize the therapeutic agents that selectively inhibit the rates of specific enzymes catalyzed processes. Enzymes kinetic thus play a critical role in drug discovery as well as elaborating the mode of action of drugs. Inhibition. The prevention of an enzyme process as a result of inhibitors with the enzyme. Inhibitors, any substance that can diminish the velocity of an enzyme catalyzed reaction is called an inhibitor. Types of inhibition. Inhibition is separated into two parts, reversible and irreversible. Reversible is again divided into competitive, uncompetitive, mixed, and non-competitive. Reversible inhibition. It is an inhibition of enzyme activity in which the inhibitor molecular entity can associate and dissociate from the protein's binding site. Types of reversible inhibition. There are 4 types. Competitive inhibition, uncompetitive inhibition, mixed inhibition, non-competitive inhibition. Competitive inhibition. In this type of inhibition, the inhibitors compete substrate for the active site. Formation of ES complex reduced while a new EI complex is formed. Examples of competitive inhibitors. Statin drug as example of competitive inhibitors. Statin drugs such as Lipitor compete with HMG-CoA substrate and inhibit the active site of HMG-CoA reductase that bring about the catalysis of cholesterol synthesis. Uncompetitive inhibitor. In this type of inhibition, inhibitor does not compete with substrate for the active site of enzyme. Instead, in another site, it is known as allosteric site. Examples of uncompetitive inhibition. Drugs to treat cases of poisoning by methanol or ethylene glycol act as an uncompetitive inhibitors. Tetramethylene sulfoxide and 3-butyl lithiolene of 1 oxide are uncompetitive inhibitors of liver alcohol dehydrogenase. So examples of irreversible inhibition. Aspirin, which targets and covalently modifies a key enzyme involved in inflammation, is an irreversible inhibitor. Suicide inhibition. It is an unusual type of irreversible inhibition where the enzyme converts the inhibitor into a reactive form in its active site. Activation. Activation is defined as the conversion of an inactive form of an enzyme to active form which processes the metabolic activity. Types of activation: activation by cofactors, conversion of an enzyme precursor. Activation by cofactors: many enzymes are activated by cofactors. Example: DNA polymerase is a holoenzyme that catalyzes the polymerization of deoxyribonucleotide into a DNA strand. It uses magnesium ion for catalytic activity. Horse liver dehydrogenase uses zinc ion for its activation. Conversion of an enzyme precursor. Specific proteolysis is a common method of activating enzymes and other proteins in biological system. Example: the generation of trypsin from trypsinogen leads to the activation of other zymogens. Enzyme specificity. Enzymes are highly specific in nature, interacting with a few substrates and catalyzing only one type of reaction. Substrate specificity is due to complete fitting of active site and substrate. Oxidoreductase do not catalyze hydrolase reactions, and hydrolase do not catalyze reactions involving oxidation and reduction. Types of enzyme specificity. Enzymes show different degree of specificity: bond specificity, group specificity, absolute specificity, optimal or stereo specificity, and dual specificity. Group specificity. In this type of specificity, the enzyme is specific not on type of bond but also to the structure surrounding it. Example: pepsin is an endopeptide enzyme that hydrolyzes central peptide bonds in which the amino groups belong to aromatic amino acids. Example: phenylalanine, tyrosine, and tryptophan. Effect of pH. The rate of almost all enzymes catalyzed reactions to the pH. Most enzymes exhibit optimal activity at pH value between 5 and 9. High or low pH value, the optimum value will cause ionization of enzyme, which results in denaturization of enzyme. Optical stereo specificity. In this type of specificity, the enzyme is not substrate but also to its optical configuration. Example: D-amino acid oxidase acts only on D amino acids. L-amino acid oxidase acts only on L amino acid. There are two types of dual specificity. The enzyme may act on one substrate by two different reaction types. Isocitrate dehydrogenase enzyme acts on isocitrate by oxidation followed by decarboxylation, two different reaction types. So till now we have seen what is enzyme, what is an enzyme, what are the structure of an enzyme, the cofactor of an enzyme, what are the types of organic cofactors, what is the substrate, what are the sites of enzyme synthesis, intracellular and extracellular enzyme, the characteristics of these enzymes, the nomenclature of the enzymes, examples for each substrates, enzymes and products, classification of enzymes, mechanisms of enzyme action, thermodynamic changes, processing at the active site, covalent catalysis, acid-base catalysis, and what is catalysis by types of catalysis, and then lock and key model, induced fit model, and enzyme kinetics. What is enzyme kinetics? What is an activation energy? What are the kinetics of enzyme catalysis? What are the factors affecting the rate of enzymes catalyst reaction? The effect of temperature, the effect of pH, pharmaceutical importance, inhibition, types of inhibition, reversible, what are the types of reversible inhibition, competitive inhibition, what are the examples of competitive inhibition, uncompetitive inhibition, what are the examples of uncompetitive inhibition, what is an activation, what are the types of an activation, activation by the cofactor, conversion of an enzyme precursor, the enzyme specificity, types of enzyme specific, group specificity, effect of pH, optimal and stereo specificity, dual specificity.